I'm David Findlay, and I'm CEO of Rostra Therapeutics, a company founded in Glasgow, Scotland in February 2022, with the specific objective of spinning out a novel platform technology from the University of Strathclyde, the Strathclyde Minor Groove Binders. Every year, over 1.5 million patients die of invasive fungal diseases. That's more than die from malaria or TB or HIV. These patients are immunocompromised and usually in hospital as a result of chemotherapy, having a solid organ transplant, having a blood condition, or even having an extended stay in the ICU. Even when treated, over 30% of patients with invasive fungal diseases die. We found that the Strathclyde Minor Groove Binders have exceptional activity against various fungi involved in invasive fungal diseases, including those resistant to current therapies. We're therefore taking forward this technology, not only because there's a high unmet need with a very high mortality rate, but also because it's an attractive commercial market. We estimate the value of the market for invasive fungal disease medications will be in excess of $12.8 billion US dollars by 2031. And every patient who is hospitalized who then goes on to get an invasive fungal disease adds a further $30,000 to the hospitalization costs. So there's a huge commercial driver and excellent potential return on investment in this space. And that's actually been demonstrated recently with a new compound that was launched back in 2015 now, called Cresemba, which has sales well over $400 million globally. And those sales have accelerated considerably year on year. Unlike many antibacterial products where we know there's a challenging and broken commercial market, the market for invasive fungal disease medications looks to be healthy and being able to offer an attractive return on investment. So that's why we chose the area of invasive fungal diseases. There's potential in other areas as well, bacteria, viruses, parasites, but that's our spearhead strategy right now. We have a fantastic team. Out of the university, we have 3 scientific co-founders. Professor Colin Suckling was a co-inventor of the Strathclyde Minor Groove Binders back in 2003. He's continued to work on them since then, as has Ian Hunter, who came into the team in 2005. He was Professor of Microbiology at the university and has also worked on those for many years. They're ably supported by Dr. Fraser Scott, who is a senior lecturer in the Pure and Applied Chemistry department at the university and has been working on these class of molecules all his academic life. On the commercial side, there's John Mulgrew, our Chief Operations Officer. John is a very experienced chemical engineer, having worked on large pharmaceutical infrastructure, brings fantastic project management and a real can-do attitude and approach to what Rostra is trying to achieve. For myself, my own perspective is that I'm big pharma through and through in a sense, except for latterly. I worked for J&J initially in sales, the sales management and product management. And then I moved into Glaxo and worked through Glaxo and then moving to SmithKline Beecham. Those 2 companies came together, so I worked for GlaxoSmithKline. And across all those companies, I was 23 years. A lot of that time spent working in the R&D organization as a commercial representative. So I would work on the investment decisions throughout the development of compounds at candidate selection, phase 1, 2, 3. I would also work on the launch plans, the target product profiles, market research, forecasts, and bring all the different commercial talent together in cross-functional teams in order to deliver excellence when bringing new products to market. A lot of that time I spent working in the antimicrobial resistance space, of course, which is an issue with antifungals as it is with antibacterials. We now have a number of compounds in early stage development. We are working closely with the NIH in the US on bringing within their preclinical services program to bring a molecule through in vitro and in vivo testing where we are now. We've been successful in getting onto that preclinical services program, as well as raising funding in the UK, primarily in Scotland, to support our scientific development. However, what we're doing now is looking to get significant private investment to help bring forward the right molecule to candidate selection, take that through pre-IND and on into early clinical development. There's been a lot of activity in this space with many big companies working with smaller companies, partnering with smaller companies. So we feel that there is definitely that commercial market, and our analysis shows that that is in fact the case. GSK have recently got involved in this space, Pfizer are involved in this space, Basilea work in this space as well. So there's certainly a lot of significant activity going on, reflecting the unmet need and the commercial opportunity. So we're seeking partners to help us on that journey, and we hope you've enjoyed what I've had to say and learned from my presentation. I'll hand over to Colin Suckling, who will take you through the scientific aspects of the Strathclyde minor groove binders.
We talk about two generations of SMGBs that are in play for therapeutic applications, and each generation contains distinct IP. The first generation is covered by a 2007 patent, and these are licensed to MGB Biopharma, which has taken one compound, MGB-BP3, successfully through a phase 2A clinical trial for the treatment of Clostridioides difficile-associated disease. This is significant because it shows that the SMGB class is druggable. MGB-BP3 does all that it says on the tin. It's rapidly effective, there's no recurrence of infection, and it's highly resilient to the generation of resistance. Now, I remember when we got the results of the phase 2A study first. After a moment of critical scientific evaluation, I realized that a compound designed by us and made in our lab had actually cured people. Now that's what we want to see, that's what we want to project into compounds commercialized through Rostra Therapeutics. And these compounds will come from the second generation, which are embodied in a 2022 patent application from the University of Strathclyde. This is going smoothly through the examination process and will be licensed to Rostra Therapeutics. The second generation compounds are an advance on the first generation because they show improvements in solubility and biological selectivity. And as you can see in the slides, the overall SMGB platform has distinct compounds that are significantly active against particular different pathogens. And as you were aware, we're looking at antifungal activity in the initial platform program for Rostra. Now, our experimental approach uses compounds that we refer to as pilot compounds. And like almost every drug discovery project, we rely upon various screens to identify our hits and leads and to profile the properties of SMGBs. But as an academic laboratory, we can't resource exhaustive synthesis and screening. So we select good quality lead compounds and treat them as pilot compounds to guide us firstly through to proof of concept in animal models of disease, and then to work our way through the detailed experiments that we'll need to take a molecule through to candidate selection. The two substantial groups of experimental data in the slide deck refer to two such compounds: a first-generation compound, SMGB363, and a second-generation compound, SMGB428. So even with limited resources, we can find out a lot about active compounds in terms of property profiles and, importantly, how to handle them experimentally. And both of these things will help us to move rapidly to candidate selection and into full preclinical development. And this is all part of a platform of SMGBs that Rostra Therapeutics will have access to. Therapies possible for many different infectious diseases can be based upon Strathclyde MGBs. For example, we have some significant possibilities in antibacterial applications, but we can't do them all, and so we're focusing on antifungal activity, as David has explained. So let's look at what SMGB363 tells us. Firstly, and most simply, this first-generation compound provides proof of concept for the treatment of aspergillosis in mice, with an activity comparable to a standard of care drug, posaconazole. But because it's a fluorescent probe compound, it also suggests important things relevant to selectivity and mechanism of action. It highlights the interaction with the fungal cell wall, which is visible through the fluorescence that you can see in several experimental results that are in the slide deck. And this makes the general point that evidence throughout the SMGB world suggests that biological selectivity is driven by selective uptake and not by selective binding to DNA. SMGB428 is a second-generation compound. It was identified as a pilot compound on the basis of its PK, which was measured at the University of Manchester. And the PK showed a surprising accumulation in lung tissues, suggesting that it would be a good compound to investigate for invasive aspergillosis. Scientists at NIH and NIAID in the USA thought so too, and we've been able to partner with them to obtain much more data about the antifungal properties of SMGB428. It stands out as being effective as a broad-spectrum antifungal with activity against azole-resistant strain, as the slide deck shows. And the necessary experiments are now in hand to push from these in vitro results to proof of concept in an efficacy model of invasive aspergillosis. Well, now we're preparing for seed investment as actively as we can. And when we get seed funding, we want to make a flying start. And from a scientific point of view, this means looking back and interrogating the screening and profiling data from our collection of SMGBs to identify promising new SMGBs for lead optimization and candidate selection. To do this, we will need to generate an efficient assay cascade with CRO partners. We will also want to consider possible downstream CMC challenges for the selected compounds. We will also want to check that the outputs of our plans will match the requirements of the regulators. And a lot of this will be helped by obtaining further data from partners about pilot compound SMGB428. And all of these things that I've just mentioned, we're now doing in consultation with our scientific advisory board members and with a range of CRO partners. So thank you for listening, and I look forward to discussing things further with you very soon.